Does the genome possess an unalterable program that determines how every human being develops from the very beginning? Or are human body cells able to adapt their genetic information to their needs? Research is helping to clarify these questions by studying the genetic material of identical twins. Why? Because their genetic material is almost identical. By the same token, there are also clearly noticeable differences, not just in physical characteristics, but also in how diseases evolve. Our genetic information is encoded in the DNA molecule. The sequence of genetic letters within it, that is, the DNA sequence, barely changes throughout a person's lifetime. In fact, it's already fixed at the point of conception. But even though each individual person's body tissue contains the same genes, the respective cells have different forms. Liver, skin, and muscle cells are clearly different from one another. The reason is that the function of a gene does not depend solely on the DNA sequence. What's also important is when and where the gene becomes active. Many genes are activated only in certain tissues and organs. Each body cell has to know when to activate what gene. It obtains that information from small changes in the hereditary material, that is, epigenetic marks. They consist of small molecules that are attached to parts of the genetic material. DNA methylation is an example of this. Enzymes attach the small methyl group directly to the genetic letters. DNA methylation can result in genes being switched off. This does not affect the sequence of genetic letters. Changes in proteins that bind to the DNA are another example. These histones act like small spindles that spool up the long DNA thread. The DNA may be packed very tightly or a little more loosely. But a gene can only become active if the DNA is packed loosely. By influencing the packing density, epigenetic marks on the histones also affect gene activity. Epigenetic marks are attained whenever body cells divide. That is how they determine the identity of the newly formed cells. Skin cells therefore only ever produce skin cells, and muscle cells only ever produce muscle cells. As an embryo develops, the epigenome ensures its orderly development and the proper functioning of its organs. Epigenetic marks are flexible, adapting to the embryo's development. The epigenome is therefore not the same in every person. Epigenetic marks continue to play an important role even once a person's body is fully developed. That's because the epigenome can also react to external stimuli and alter the activity of certain genes. In this way, the cells and the body are able to adapt to the environment. Epigenetic marks also influence the way in which diseases evolve. This was demonstrated in identical twins, where one twin suffered from type 1 diabetes. Clear differences were noted in the epigenome of the diabetic twin and the healthy twin. Such differences point to environmental influences that possibly may favor the development of the disease. Can epigenetic mutations also be passed on to offspring? In worms and plants, it is possible. But in humans and other mammals, there is a particularity in that, during reproduction, epigenetic marks in the nuclei are erased. It is still unclear whether subsequent generations nonetheless inherit at least part of the genetic information concerned.